Now, let us look at the female reproductive parts of the flower, that is the gynoecium. What does this gynoecium consist of? We learned that androecium consists of stamens. Androecium consists of stamens. Whereas gynoecium consists of pistil. If the gynoecium, it contains only one pistil, single pistil, it is called as a monocarpellary. Mono means single, only one. But if the gynoecium, it contains so many pistils, it is called multicarpellary. Multi means many. Multicarpellary. There are many carpels in that flower. So gynoecium is of two types. One, monocarpellary, that means with single pistil, and multicarpellary, that is with many pistils. Let us look at the different parts of the pistil. So this is the pistil, and here it has got three important parts. What are the three important parts? One, stigma, and two, style, and the third one is ovary. Stigma, style, and ovary. So the stigma is the landing platform. Landing platform for what? Pollen grains. Pollen grains, they come and stand on the stigma. They stick to the stigma. So the stigma is sticky. So by that it receives the pollen sticks to the stigma, and from there it germinates. The pollen germinates on the stigma and they produce pollen tubes. So the pollen tubes that are produced by the pollen grains, they enter the next part of the pistil, that is the style. Style is the elongated and slender part through which the pollen tube passes. So the pollen grain from the stigma, it produces a pollen tube which passes through this style, and finally it reaches the third part, that is ovary. Ovary is the basal bulged part. It is bulged. It has got ovules and other parts in it. It is bulged. So in this way the pollen tube, it travels and it reaches the ovary. So these are the three main parts of the pistil. What are they? Stigma, style, and ovary. So now we see the two different types of multicarpellary gynoecium. So gynoecium is two types we learned, monocarpellary and multicarpellary. Now this multicarpellary is of two types, syncarpous, apocarpous. What is this syncarpous? Syncarpous is a type of gynoecium in which pistils fuse together. So there are so many pistils. So that's why it is called as multicarpellary. And all these pistils are fused together. If a flower contains many pistils, and if all the pistils are fused together, we call it as syncarpous. Syncarpous. Example, papaver, poppy plant. This is the poppy pod we can see. This is the poppy flower. Here we can see the fused pistils. The next type is apocarpous. Apocarpous means the pistils are free. They are free. Here also many pistils are there, but they are free. Here we can see the free pistils. The pistils are free. And here the example is Michaelia. So this is about the types of gynoecium. Now, let us look at the parts of the ovary. So this is ovary, and the ovary consists of ovarian cavity. The cavity present inside the ovary is called ovarian cavity. It is also called as locule. Locule. So inside this ovarian cavity, we find a tissue called placenta. Placenta is the tissue from which the ovules are formed. Ovules are called megasporangium. Megasporangium is formed from placenta. So these ovules, they consist of embryo sac, and this embryo sac, it consists of egg cell and other cells. So in some plants, the pistil, it contains only one ovary, single ovary. Examples, wheat, paddy, and mango. But in some plants, the pistil contains many ovaries. Many ovaries are present in single pistil. Example, papaya, watermelon, orchids. Now, let us look at the structure of the microsporangium, that is the ovule, the structure of the ovule. So this is the structure of the ovule. Let us see its parts. The first part is funicle. Funicle. So this is the funicle. It is a stalk which attaches the ovule to placenta. So this is the ovule. Ovule is attached to placenta. So this is placenta. So to this placenta, the ovule is attached with the help of a stalk called as funicle. And the next part is hilum. Hilum. This is hilum. So this hilum, it is the bridge between the funicle and ovule. So this hilum, it is the bridge between funicle and ovule. That is hilum. And next part, integuments. Integuments. Outer integument, inner integument. So here, this is the outer integument. These two are outer integuments. And these two are inner integuments. Inner integuments. So the ones which are highlighted in pink are outer integument, and the ones which are highlighted in blue are inner integuments. So what are these integuments? They are the protective envelopes. They are the protective envelopes. So they protect the inner nucellus and embryo sac. So the inner parts are nucellus and embryo sac. These two are protected by the outer and inner integuments. Now the next part, that is nucellus. Nucellus. So this area is nucellus. So this area is nucellus, which is covered and protected by the inner and outer integuments. So the nucellus, what does it contain? It contains the reserve food materials. It contains the reserve food materials that are necessary for the development of embryo sac. So the center one is the embryo sac. So this central part is called as the embryo sac. So the embryo sac is covered by nucellus. Nucellus, it is the reserve food material, and the nucellus is protected by inner integument and outer integuments. These integuments, they leave a gap at this place. We can see the integuments are covering everywhere, all around, but except this area is left free. So this area is called as micropyle. And why this is left free? Why the integuments are not covering the nucellus at this area? That is for the entry of pollen tube. So the pollen tube, it enters the ovule through this gap, and it carries the male gametes into the embryo sac. So the end of the ovule, which is towards the micropyle, it is micropylar end. It is micropylar end. And the end of the ovule towards the basal side, it is called as chalazal end. Chalazal. This end of the ovule is chalazal end, and this end of the ovule is micropylar end. So what are the different parts of the ovule? Funicle, hilum, outer integument, inner integument, nucellus, embryo sac. So these are the different parts of the megasporangium or ovule. Now let us look at the formation of megaspores from the megaspore mother cell. So this process is called as megasporogenesis. What is this megasporogenesis? The process of formation of megaspores from the megaspore mother cell. Megaspore mother cell. So what are formed? Megaspores are formed from the megaspore mother cell is called megasporogenesis. This process is called as megasporogenesis. Right. So megaspores are the embryo sacs. Megaspores are the embryo sacs. So this embryo sac is formed from the megaspore mother cell. So where is the megaspore mother cell? Here we can see. So here, this area is the nucellus. This is the nucellus. So this highlighted area is the nucellus. So here we have taken the cells of the nucellus. Here we can see the nucellus here. And these are the cells. And one of the cell of the nucellus, it differentiates. It develops into megaspore mother cell. Megaspore mother cell. It develops into megaspore mother cell. This is the megaspore mother cell. We can see this megaspore mother cell is very big. It is large in size. And inside the cell, we see so many dots. We see so many dots. So the center one is the nucleus. It has got a big nucleus. So the cell that is formed from the nucellus is large in size. It is a large cell and it has got a prominent nucleus. See the nucleus? It is big nucleus and dense cytoplasm. The cytoplasm has got so many dots. It is dense cytoplasm. So what are the features of this megaspore mother cell? It is large in size, it has got a prominent nucleus, and it is having dense cytoplasm. These are the features of megaspore mother cell. So from where does this megaspore mother cell has come? It has come from the one of the cells of the nucellus differentiates into megaspore mother cell. From this megaspore mother cell, what will be formed? Megaspore is formed. So that process is called megasporogenesis, formation of megaspores from the megaspore mother cell. So now here, this particular cell, the big cell, it undergoes meiotic divisions. Meiosis takes place. So in the meiotic divisions, it becomes into two cells. 1, 2. Two cells. So it is called as megaspore dyad. Dyad means two cells. 1, 2. Two cells. One big cell, megaspore mother cell, divided into two cells. So again, the divisions continue and the two cells become four cells. Here we can see 1, 2, 3, 4 cells. Four cells are formed. So these four cells are called megaspore tetrad. Megaspore tetrad. Here it is called megaspore dyad. Two cells, four cells, megaspore tetrad. So all these four cells are haploid cells, haploid megaspores. So these four megaspores are not going to develop into embryo sac. All these four cells are not going to develop into embryo sac. Only one. So only one survives and three degenerates. Three cells degenerate, only one survives for the further process. So this is how the microspore tetrad is developed from the megaspore mother cell. So now let us see the development of embryo sac from the megaspore, right? So we have seen that four megaspores are formed, 1, 2, 3, 4. Out of this, 3 they will disintegrate, disappear, and 1 remains. So the 1, it develops into a functional megaspore. It develops into a functional megaspore. So this single functional megaspore, it has got only one nucleus. Here we can see one nucleus, and it undergoes mitosis, and here it formed a 2-nucleate embryo sac. 2-nucleate embryo sac is formed. So this is only one cell, but 2 nuclei are there, 1, 2. Again, by mitosis, it develops into 4-nucleate embryo sac. So 4-nucleate, 1, 2, 3, 4. 4-nucleate embryo sac. And still the mitosis continues and 8-nucleate embryo sac is formed. 8-nucleate embryo sac. Here we can observe that only the nuclei are being divided, not the cell. So the nucleus is not surrounded by cell walls and new cells are not formed. Only the nuclei is getting divided and finally 8 nuclei are formed. Only one cell. Within one embryo sac, 8 nuclei are formed by mitotic divisions. So this kind of division is called as a free nuclear division. Free nuclear division means only the nuclear division takes place. Only the nuclear division, no cell wall formation. So here we got an embryo sac with 8 nuclei. Now the formation of cell walls takes place. So here, out of these 8 nuclei that are formed in the embryo sac, single cell, they get the cell walls and they form the cells. 3 of the nuclei, they form the antipodals. 3 antipodals. So these antipodals, they move towards the chalazal end. This is the chalazal end of the embryo sac and this is the micropylar end of the embryo sac. So here we can see towards the micropylar end, the synergids are formed and egg cell is formed. Synergids and egg cell. 2 synergids are formed and 1 egg cell is formed. So total 3 cells are formed towards the micropylar end. And towards the chalazal end, 3 antipodals are formed. So here 3 and here 3, total 6. And this is all one cell, one big cell here. This is all one cell, right? So this cell is called central cell. Now the central cell has got 2 nuclei, 1 and 2. So these 2 nuclei of the central cell are called polar nuclei. Polar nuclei. So total 8 nuclei. They formed the 2 synergids, 1 egg cell, 1 central cell and 3 antipodals. So it develops into the embryo sac. Now let us look at the different parts of the developed embryo sac. Here, this is the completely developed embryo sac. Here we can see this is the central cell. This is all the central cell. It has got 2 nuclei. These are the polar nuclei, and it has got 3 antipodals which are towards the chelazal end. Here we have written the chelazal end this side. Now we made this diagram upside down. Here this is the chelazal end and this is the micropylar end. And here it has got synergids, 2 synergids, 1, 2, and this is the egg cell. And this is the central cell. Now the male gametes that come from the pollen grain, they have to enter and reach the egg cell. So how do they enter? From which side they enter? From the chelazal end or from the micropylar end? They enter from this micropylar end. So this filiform apparatus, whatever the filiform apparatus are there, these filiform apparatus, they facilitate the movement of pollen tube into this embryo sac. So they help for the movement of entry of the pollen tube. So the pollen grains enter into this embryo sac at this end, micropylar end. So this egg apparatus, the synergids and egg cell, these 2 synergids and egg cell are called egg apparatus. Egg apparatus means 2 synergids and 1 egg cell. So if one embryo sac is developed from a single megaspore, it is called monosporic development. Here only one embryo sac is developed. Here only one. Out of these 4 megaspores, 3 degenerated, only one is formed into a functional megaspore which developed into an embryo sac, and this kind of development is called as monosporic development. So far we have seen the andrecium, that is the male reproductive part, and gynoecium, the female reproductive part, the development of microspores and megaspores. Now let us see the process of pollination. What is pollination? We already learned this in class 10. Transfer of pollen grains to the stigma of a pistil is termed as pollination. So this is the stigma and this is the anther. So these are the pollen grains. The pollen grains, they transfer from anther to stigma. This process is called as pollination. So there are different types of pollination. Let's say kinds of pollination. What are the different kinds of pollination? Here 3 kinds are discussed here. 1, autogamy, and 2, gatonogamy, 3, xenogamy. Let's see what is this autogamy. Autogamy means if the pollination takes place within the same flower, it is called as autogamy. Here we can see that inside the same flower, here stamens are there, at the same time pistil is also present. If the pollen grains of the same flower, they reaches the stigma of the same flower, it is called as autogamy. So here, for this autogamy, certain conditions are required. Only then the autogamy is possible. Otherwise, autogamy is not possible. See, if stamens and pistils are exposed completely, then autogamy is not possible. That means if the stamens and pistils are completely exposed, means the air can carry some pollen from other flowers. The insects may carry the pollen from other flowers. So autogamy is not possible. Cross-pollination takes place. So for the plants in which autogamy is required, in that plants, the stamens and pistils should not be exposed completely. They should be closed. And next one, pollen release and stigma receptivity in synchrony. For autogamy, the pollen release and stigma receptivity means the receiving capacity of the stigma, the readiness of the stigma and release of pollen should be done at the same time. Then only autogamy is possible. If pollen is released and stigma is not ready to receive, then autogamy is not possible. They should be in a sync. pollen release and stigma receptivity, they should be in a synchrony. And next, the anthers and stigma should be close. They should be close. They should be side by side. Then there is a possibility for autogamy, right? So next one, geitonogamy. Here in this case, anther to stigma of another flower of the same plant. What is this geitonogamy? Anther to stigma of the another flower of the same plant. This is also a type of self-pollination. This looks like cross-pollination, but it is also a kind of self-pollination, but it happens between two different flowers of the same plant. Same plant. So in this case, in case of this geitonogamy, genetic similarity. We see the genetic similarity. That means there is no change in the genes, no variations, because they both belong to same plant. Right? But not within the same flower. Two different flowers of the same plant. It involves a pollinating agent. In case of this geitonogamy, the pollinating agents are required. Like some wind or insects should carry the pollen from this particular flower to this particular flower. It is carried by some pollination agent, or pollinating agents are required. Okay. So that is geitonogamy. Next we see xenogamy. Xenogamy, anther to stigma of a different plant. So this is one plant, this is another plant. Here the transfer of pollen from one flower to another flower of different plants takes place. This is called as xenogamy. Okay. So this is the true cross-pollination. So why we are calling this as true cross-pollination? Because there is genetic diversity. So this plant is different, this plant is different, so there is genetic variation. Right? This is xenogamy. In some plants, there will be two different kinds of flowers. Let's see. So in some plants like viola, oxalis and commelina, in these plants, the plants, they bear two different types of flowers. So here is the example of commelina. This is the commelina plant. So now this plant has got two types of flowers. Here we can see one type of aerial flowers, the flowers which are present outside the ground, that is present on the stem, aerial flowers. These flowers, they have exposed anthers and stigma. These flowers are called as chesmogamous flowers. They have exposed anthers and stigma. These are the aerial flowers. The same plant has got another type of flowers. Here we can see under the ground, cleistogamous flowers. So this is a cleistogamous flower. So these cleistogamous flowers, they do not open at all. So where are the anthers and stigma, anthers and the pistil? They are present inside. They are not exposed. And in such cases, in case of these cleistogamous flowers, only the autogamy takes place. In these flowers. There is no chance for geitonogamy. There is no chance for xenogamy. Why? Because the reproductive parts are not exposed. So no pollen can enter the flower from outside because it's completely closed. Whereas in case of chesmogamous flowers, they are open. So there is a chance of cross-fertilization. So both type of flowers are present in these plants. Examples are viola, oxalis and commelina. So this is about these types of pollination. Next we see the types of pollinators, the agents of pollination. Now let us look at the agents of pollination. So there are two different types of agents of pollination. One is abiotic and the other one is biotic. So biotic means insects, animals which contribute to the pollination, or the biotic agents of the pollination. But most of the pollination is done by the abiotic factors. What are the abiotic factors? Wind and water. These two are the abiotic agents of pollination. Most of the pollination is done by these two, wind and water. Only a small portion is done by the biotic agents. So we'll see about these biotic agents later. Now let us see about these abiotic agents, wind and water. So in case of abiotic agents of pollination, there are less chances of contact. Less chances of contact of what? Of pollen and stigma. Chances are less because they are carried by the wind and water. So the certainty is very less. It is not very sure that pollen reaches the stigma because the air has to carry, the wind has to carry. It is not in any particular control. So it is by random chance the wind carries the pollen. If the pollen reaches the right stigma, then if the stigma receives that pollen, then the pollination takes place. So the chances of pollination are very less in case of these abiotic factors like wind and water. And next one is enormous pollen. Enormous pollen is produced compared to no number of ovules available. Enormous pollen is produced compared to number of ovules available. Ovules available will be less. More amount of pollen is produced. Here in this picture we can see that the pollen is looking like a dust, like a smoke. Large amount of pollen is produced. Because the chances are very less. The chance of contact is very less. So large amount of pollen is produced. Out of this large amount, only a few will reach the appropriate stigma for pollination. So when we compare the amount of pollen released and amount of ovules available, the ovules will be available in less quantity, but large number of pollen has to be produced for the case of wind and water. And in this abiotic agents, in case of these abiotic agents where the pollination is done by abiotic agents, the flowers are not attractive. Flowers are attractive. Flowers are fragrant for insects and birds because insects and birds are attracted by colorful flowers, scented flowers. But here the pollination is done by wind and water. There is no need of colors, no need of scents and attraction. So in case of these abiotic agents, flowers are not attractive. Now let us see pollination by wind. So this is the most common type compared to water. Most of the pollination is done by the wind when we compare wind and water. In case of water, it is very rare. Only a few plants. Only in case of a few plants, the pollination will be done by water. And here, the pollen grains are light and non-sticky. In case of water, pollen grains are light and non-sticky. Why they are light? Because they should be easily carried away by the wind. If they are heavy, due to gravity, they cannot be carried to long distances. And they are non-sticky. If they are sticky, they stick to the surfaces. They cannot be taken to the stigma. They cannot be carried by the wind if they are sticky. So they are non-sticky and light. And they have well exposed stamens. The flowers that undergo this pollination by the wind, those flowers will have well exposed stamens. So when the stamens are well exposed, then the pollen can be easily carried away by the wind. And these flowers, they have another feature, that is feathery stigma. The stigma is feathery. Here we can see that the stigma is feathery. So many feather-like structures are present in the stigmas. So why they have stigma, feathery stigma? To catch the pollen, to trap the pollen. That is to trap the pollen. And most of these flowers will have only one ovule in the ovary, and they'll be having inflorescence. Most of the flowers in which the pollination done by wind, they have inflorescence, bunch of flowers. And examples, corn cobs, and the tassels of the corn cobs. So we know the corn cob. So the corn cobs, they will have tassels, some hair-like structure. If you see that regular corn or sweet corn, you can see that at the top you will find some hair-like structure. These hair-like structures are called as tassels of corn cob. These are nothing but the stigma and the style of the flower. Why they are outside? They are outside the pod because to get the pollen capture, right? So the reproductive parts, the stigma, style or anthers, are exposed well in case of flowers which usually get pollinated by the wind. And this kind of wind pollination is most common in grasses. In grasses, we see this kind of wind pollination. Now let us see the water pollination. So this is very rare. It is limited only to 30 genera, and in most of the monocots it takes place, and it also takes place in algae, bryophytes and pteridophytes. The pollination by water takes place. So in the water pollination, we can observe this pollination by water in fresh water as well as in marine water. So example for freshwater, Vallisneria and Hydrilla. In these plants, pollination will be done by water. And in case of marine, the example is zoster. In case of zoster plants, pollination will be done by water. So some water plants, they do not get pollinated by water. For example, water hyacinth and water lily. So we see these plants in very large quantity in ponds and lakes, water hyacinth and water lily. They do not get pollinated by water. So they'll have their flowers outside the water, usually get pollinated by the wind or insects or other means, not by water. So here in this picture, we can see the Vallisneria. We can see the Vallisneria plant. So the plant is having the flower above the surface of the water. We can see that it is above the surface of the water. So the female flower, it comes to the surface of the water, and the male flowers or pollen grains, they travel on the surface of the water and they reach the female flower. So in some cases of water pollination, it is done on the surface. Example, Vallisneria, surface of the water, not inside the water. In some cases, the pollination will be done inside the water. The female flower will be inside the water, and even the male flower, the pollen grains are also released inside the water. In such cases, the pollen grain, there is a chance for the pollen grains to get wet. They may become wet. So the pollen grains may become wet. So to avoid that, to protect the pollen grains from getting wet, so to protect the pollen grains, mucilaginous covering will be present on the pollen grain to protect it from wetting. So in which case, in the cases where the pollen grains, they travel inside the water, the pollination that takes place inside the water. But in case of Vallisneria, it takes place on the surface of the water. So this is about the abiotic agents of pollination. Next we see and we learn about the biotic agents of pollination. So now let us look at the biotic agents of pollination. So the biotic agents of pollination, what are the different biotic agents? Here, insects, small animals and large animals. So the pollination by large animals is very less, and the pollination by insects is more compared to the other animals, right? So here, what are the different insects by which the pollination takes place is bees, butterflies, flies, beetles, wasps, ants and moths. These are the various insects that help in the pollination. So whereas in the small animals like sunbirds, hummingbirds and bats, here we can see the birds. So these small animals also contribute for the pollination, and large animals like lemurs, rodents, gecko lizards, garden lizards, and even sometimes monkeys. So all these animals, they help in the pollination. So these are the different animals that takes place in the pollination. So the flowers in which the pollination is done by the biotic factors like insects and small animals, they have certain features, special features. Let us see what are those. So the features of insect pollinated flowers, they are large in size. Insect pollinated flowers are large and they are colorful, they are fragrant, they have good colors, bright colors and good smell, fragrance. So by that most of the insects are attracted because of the color and the smell and the size. And they are rich in nectar. So nectar is the reward, nectar and pollen. These two are rewards for the insects and animals. So they come to the flowers for this nectar. So the flies and beetles, In case of flies and beetles, the flowers will have foul odor. In case of other insects, they have fragrant smell. But flies and beetles, they go to the foul odor. So certain flowers which are pollinated by the insects, like flies and beetles, these flowers, they'll have the foul odor because the flies are attracted to the foul odor. So in this case, the pollen gets coated to the animal body. So here we can see when the bee, it visits a flower, the body of the bee is coated by pollen grains. So large amount of pollen grain sticks to the body of the bee. When the bee visits the next flower, the pollen grain drops on the next flower stigma and get pollinated. In some cases, in most of the cases for all these insects, the rewards are nectar and pollen. For the birds also, the nectar and pollen are the rewards. For the rewards, the animals, they come from the flowers and they help in pollination. But in some cases, in special cases, there is a reward like safe place to lay eggs. This is also a type of reward. So here there is a relation between yucca plant and moth. Moth, it's a kind of insect. Here we can see the moth. You can see that the moth is laying the eggs inside the flower. Inside the flower. So the moth is laying the eggs inside the flower. That means what is the reward here? Safe place to lay the eggs. Once the eggs become into larva, they come out of the flower. But how this is beneficial to the flower? Pollination. Pollination is done by this moth. And the flower, it provides a safe place for the moth to lay the eggs. So in this relation also, this kind of pollination takes place. So the moth deposits the eggs in the locules of the flower. So here examples are Amorphophallus and Yucca. These are the examples for the flowers which provide a safe place to lay the eggs. So these are the features of insect pollinated flowers, and these are the different biotic agents of pollination: insects, small animals and large animals. Thanks for watching. Please like the video. Please share this video with your friends. Please subscribe to Grade Booster channel. Press the bell icon to get all the latest updates. Check the description